Today I'm going to talk to you about our recent research on GzmK and its role in promoting IL-23 secretion as well as keratinocyte proliferation in psoriasis. So GzmK is a serine protease with tryptase-like activity. It was first discovered within the cytotoxic granules of cytotoxic lymphocytes as well as natural killer cells, along with the remaining Granzyme family members. And because of this, Granzymes were long thought to function exclusively in immune cell-mediated killing. And we know that with the help of perforin, the pore-forming molecule, GzmK can be internalized into target cells, and here it can cleave a variety of intracellular substrates that have implications in cell death as well as viral replication. However, more excitingly, in recent years, we've shown that Granzymes, including GzmK, can accumulate in the extracellular milieu, both from leakage of the immunological synapse as well as the secretion by cells, immune cells that do not express perforin, as well as non-immune cells. And within the extracellular milieu, GzmK, we are learning, has a role in a diverse amount of functions. This is through the cleavage of extracellular substrates, many of which have still yet to be identified, but perhaps the most well-established is the cleavage and activation of protease-activated receptors. In protease-activated receptor 1 in particular, cleavage and activation by GzmK has been shown to induce the release of various pro-inflammatory cytokines as well as proliferation of various cell types. And so as both GzmK and PAR1 levels are elevated in psoriasis, and the fact that GzmK-mediated PAR1 activation is linked to both proliferation and inflammation, which are two disease hallmarks of psoriasis, we hypothesize that GzmK might mediate keratinocyte proliferation as well as inflammation in psoriasis through PAR1-dependent mechanisms. And so first, to assess whether GzmK plays a role in disease development, we use the well-established imiquimod mouse model of transient disease, looking for phenotypic differences between wild-type and GzmK knockout mice. We scored the severity of the mice throughout the duration of the study using a modified version of the Psoriasis Area and Severity Index. And then at termination, we collect skin tissues and look for what are the largest differences in disease hallmarks between these two genotypes. And what we observed is that although there are no morphological differences in our healthy GzmK knockout mice compared to that of the wild type, the GzmK depletion makes these mice less susceptible to psoriasis-like skin development in this model. So in particular, disease severity, which was defined as cumulative erythema as well as desquamation, was significantly decreased in the GzmK knockout mice compared to the wild type at day 6 and 7 of this trial, and this difference was magnified over this period of time. What's more, when we look to the histology, we see that the largest disease hallmarks that had differences between the genotypes were that related to epidermal thickness and epidermal hyperplasia, as well as the influx of immune cells and levels of pro-inflammatory cytokines, which each of these being significantly decreased in the knockout mice compared to that of the wild type. And so next, we wanted to confirm whether these findings can then translate back into human cells, and if so, what are the putative molecular mechanisms, and might they be dependent on GzmK's ability to cleave and activate PAR1? So we cultured human keratinocytes in vitro, and these were transfected with or without an siRNA targeting PAR1, and then we stimulated them with GzmK and measured proliferation in the form of immunocytochemistry for Ki67, as well as cell counts. And what we saw is that in those cells in which PAR1 is not affected, that GzmK is able to induce the levels of Ki67, as well as we saw increased cell count numbers. However, when PAR1 is knocked down, we see that this effect is completely abolished. So in fact, GzmK is able to stimulate human keratinocyte proliferation, and this occurs via a PAR1-dependent mechanism. Now we were interested in seeing what the downstream intracellular signaling pathway might be involved. In particular, we are interested in looking at MAP kinase and STAT3 phosphorylation, as both of these have been implicated in keratinocyte proliferation in the context of psoriasis, as well as we know from previous work that GzmK cleavage and activation of PAR1 can induce the phosphorylation of P44-42 MAP kinase. And what we see, again, in the keratinocytes in which PAR1 is not affected, we see Granzyme K is able to phosphorylate P44/42 MAPK as well as STAT3 as measured over the total forms of the protein. However, when PAR1 is knocked down, this inhibits Granzyme K's ability to phosphorylate these proteins. And following this, we wanted to see in particular whether the Granzyme K phosphorylation of STAT3 was occurring directly downstream of P44/42 MAPK phosphorylation or via a different signaling pathway. And so we performed a similar experiment, but this time pre-treating cells with a MAPK inhibitor prior to Granzyme K stimulation, and then looking to see whether Granzyme K lost its ability to phosphorylate STAT3, and that is in fact what we did see. So this tells us that the phosphorylation of STAT3 by Granzyme K is dependent on the phosphorylation of upstream P44/42 MAPK, and of course both these events being dependent on PAR1 activation. Now, on a slightly different note, I touched on before that we've also showed that Granzyme K can mediate inflammation. In our mouse model, we saw large differences in terms of the numbers of immune cells as well as pro-inflammatory cytokines, with these being elevated in the wild-type mice compared to that of the knockout. One of the standout pro-inflammatory cytokines, which we saw the largest differences in, was IL-23. And so we raised the question whether Granzyme K might have a direct role in inducing the secretion of IL-23 from cells. And in the context of macrophages, we were able to show that when stimulated with Granzyme K, these cells produce increased levels and secrete increased levels of IL-23. And this was true for both M0 or a resting state macrophage as well as M1 or a pro-inflammatory state macrophage. And we have some preliminary evidence to suggest that this might be occurring by a similar mechanism to that of the keratinocytes. However, this is still yet to be confirmed. So in summary, we know that Granzyme K can cleave and activate PAR1 on the cell surface. And this results in irreversible binding of the G partner protein, which leads to successive downstream signaling events. In the context of keratinocytes, we've been able to show that Granzyme K can phosphorylate the MAP kinase P44/42 and successive STAT3. STAT3 being a transcription factor which has implications in psoriasis, particularly in the survival and proliferation of keratinocytes, as well as immune regulation. And so we believe this might be a putative mechanism by which Granzyme K can increase psoriasis disease severity, as we saw in our murine model of disease. Altogether indicating Granzyme K as a promising therapeutic target for psoriasis, and we're hopeful that these continued discussions will provide new avenues for drug discovery and help to further refine our understanding of disease mechanisms. And we're currently in the final stages of review prior to publication for this work, so hopefully it'll be available for you to read soon. At this time, I'd just like to thank my lab members, funders, and collaborators with whom this support. This work could not be made possible, and a special thank you to the International Psoriasis Council for having me speak today.
Thank you. So maybe time for one quick question. So I have one quick question. So besides macrophages, have you looked into whether any other minor cells would have that effect?
Whether Granzyme K can induce IL-23 from other cells? Yeah, we did look at dendritic cells. However, we did not see the same results here, but it could be due to the fact that the model of dendritic cells we're using, because we didn't use a primary, we're using an immortalized cell line that's then differentiated into dendritic cells, that maybe that's the cause for the discrepancy in what we're seeing.
Okay, interesting.
Just one quick. Just one. Well, I'm stupid. The source will be fibroblasts in that regard of Granzyme K?
The source of Granzyme K?
Yeah.
In our psoriasis lesions that we looked at, it seems that the primary source is inflammatory M1 macrophages as well as mast cells. And then there's some T cells too.
And you think that they then activate the basal keratinocytes to produce IL-23?
Yes, and that's our hypothesis. And then when in the immunohistochemical staining, we see that the cells are mainly present just under the basal keratinocytes. So we think they might be able to, activate them and promote the proliferation.
To ask Alex and Johan for the spatial.
Yeah.
Sure.
It was very interesting. Thank you.
All right, thank you.